Hey guys, welcome back to another lesson. So this lesson is going to be a very high yield lesson when it comes to the translation. The translation process is a very important process that occurs inside our cell when it comes to the synthesis of the polypeptide that we are going to use and become a protein. The thing is, for the IMAT and the BMAT exam, what I noticed is that it can be very easy to study a lot of unnecessary things when it comes to the translation and the transcription. Because there are so many things called transcription factors, which are proteins that help us along the way, and coenzymes that intervene and take action along the way. And it's so unnecessary to study them because at the end of the day, it's very important to study the highest yield points for the IMAT exam. So for the IMAT and BMAT exam, when it comes to translation, here are the three things, the main things, the processes you need to know. The process of translation starts in something called initiation. Initiation is when you have the complex of the small subunit, I'm talking about eukaryotic cells, the small subunit with the start codon, that the start tRNA that attached to methionine, which is going to recognize AUG, which is the start codon, and it is going to be attached to the guanosine, the guanine, modified guanine cap that we have, and we talked about when we talked about the processing of the mRNA, the pre-mRNA, when it comes to eukaryotic cells. Then it is going to start slide from the 5 to the 3 prime, all the way until it actually finds the AUG start codon. Once it finds the AUG start codon, you will see that the ribosome is able to start recognizing more codons as well. And we will have the process of elongation. Elongation is where you have between the start and stop codon the formation of peptide bonds between many different amino acids when the tRNA are going to be inserted inside the ribosomes and the formation is going to occur. Then it's going to slide all the way from the AUG to the stop codon, and you will see that the stop codon doesn't contain any amino acid. It is not associated with any amino acid. It is associated with something called the releasing factor. And once the releasing factor is inside the ribosome, the polypeptide is going to be released. And then the ribosome is going to be released to and detached from the mRNA strand, and then you will see that this mRNA strand is freely available now for another round of translation. This process happened multiple times until the natural, either the natural dissociation and the breakdown of the RNA molecule, or different enzymes that catalyze the breakdown of this RNA molecule. The nucleotides of the RNA are going to be, of course, recycled, and then you are going to form new RNA when you do the transcription and the translation, and the cycle goes on and on and on. Now, the process of terminating and reaching the stop codon and getting this releasing factor inside the ribosome to release the polypeptide is called termination. So we have the process of 1. Initiation, 2. Elongation, and 3. Termination. And you do it over and over and over and over and over again. Now, I found this nice GIF online that is going to show you exactly what we need to study when it comes to the elongation process of the mRNA. So we have 3 main areas inside our ribosome. We are going to have the E area, we are going to have the P area, and we are going to have the A area. Now, the following letters are just an abbreviation. They are symbols, they symbolize the following thing. 1 stands for exit, P stems from peptide, for the peptide that it holds, because the polypeptide chain that is going to be produced is going to be holding the P site, and the S sides is for the amino acid. The full name is also amino acyl tRNA transferase, which is the enzyme that is actually being used and that you don't need to actually memorize the names. Just remember that the A site is the area that the new tRNA is going to be inserted. As you can see here, it is going to be inserted with the new freely available tRNA. The P site is going to hold our polypeptide chain. Between the P and the A site, you are going to have the peptide form, and once the ribosome is going to move 3 codons and continue to, 3 nucleotides and continue 1 codon, it is going to, the tRNA is going to be inserted inside this E site and is going to be released and removed from the ribosome. So the cycle is as the following. tRNA will come and be inserted into the codon that is next to the P area with a new amino acid. Then you will see the formation of peptide bond between the two tRNAs, amino acids, that are found in the P sites and the A site. Then you will see the elongation and the formation of polypeptide chain in the P site. And once the ribosome itself moves to the right from the 5' to the 3' end to the next codon, the A site will accept a new tRNA with a new amino acid, but the P site amino acid that was pushed to the E site is going to be released and removed from the ribosome. And this cycle is going to repeat until we reach the stop codon. Once we hit the stop codon, instead of this tRNA, we are going to have something inside our A site that is going to called a releasing factor. And the releasing factor is going to help us, one, disassociate this formation between the tRNA and the amino acid, and two, because we don't have any amino acid, it is going to release the polypeptide because now we don't have any peptide form to form. So inside of this tRNA, you are going to have an empty space with a releasing factor, and the releasing factor is going to be assigned to the stop codons, and then you have the release of the polypeptide. Then the small and the big and the small subunits are going to be released from the mRNA, and the mRNA in the ribosomes are going to be recycled and to form again. The ribosomes are not going to be consumed during the process. The mRNA is not going to be consumed during the process. The only thing that is being consumed and break down is the linkage between the tRNA and the amino acids in order to form new linkage, new peptide bond between the amino acids that you see in the P site, that forming the following chain. And this is basically everything you need to know for the IMAT and the BMAT exams when it comes to the formation of polypeptide chain inside the ribosome. So if I sum this video up, you have the E space where E zone where it stands from exit, you have the P for peptide and A for amino acids. The A will be next to the P, and the A will accept new tRNAs that are attached to an amino acid. This area, if we get the wrong tRNA, the wrong codons, it will just spit out the wrong tRNA and wait for the correct tRNA to join. Once we have the correct tRNA inside this area that fits the codons, the anticodon are going to fit the codons, we are going to have the peptide bond formation between the tRNA in the A site to the tRNA in the P site. Once we have this peptide bond formed, the ribosome is going to skip and to go to the next codon. It's going to go to the next codon. Therefore, the tRNA in the P site is going to be pushed to the E site and is going to be removed. And the tRNA that was in the A site is going to be pushed to the P site and continue the chain of amino acids. I hope this video made sense, and I will see you next time.